Hello, good morning. Today we're going to look at testis and spermatogenic cells. This is part of the Advanced Microscopic Anatomy course. So we're going to appreciate the microscopic features. This class is not basically for you to start relearning what you've learned. It's actually for you to focus on what matters in at the advanced stage. So you want to focus on the microscopic features of the testis and the spermatogenic process. I mean, spermatogenesis as a whole, both the spermatocytogenesis and the spermiogenesis. So I'm just going to highlight what I I feel is very important. I'm going to focus on the spermatogenic cells and then focus more on the testis. Of course, when you hear testis, what comes to mind is basically, a functional reproductive organ for the male where the production of the semen or rather sperm is being concentrated. Okay, is responsible for production of spermatozoa and also generates other components of the semen as well with other accessory organs of the reproductive system. So this is the picture of that. You can see this is the appreciation of the gross nature of the testis. So you can see just parallel to it, more like a superimposed anteriorly located epididymis is there. And then you're going to appreciate the ductus deferens, that is what you call the vas deferens, that is responsible for the transportation of the sperm, when it is being ready for ejaculation. So when it's being produced, after being produced in the testis, it's being stored in epididymis to give it some level of motility, and then it moves through the vas deferens. And of course, at this point, you have other fluids coming in from seminal vesicle, from ovular glands, and then before uh, ejaculation through the urethra opening. Okay, so let's look at more intimately at other things that make up, as a way to introduce the testis. It's the I've talked about being the major and productive organ of the male, just like you have the ovary for the female. Okay, it lies within a serous sac that is called a tunica vaginalis. Actually, there are three layers of sac which we are going to see, but basically, the testis lies in a very serous, highly serous sac that is called a tunica vaginalis, and each of these testes measures around 4 cm in terms of length. It is also oval in shape. Okay, so that tunica albuginea is part of more like the external part of the tunica vaginalis, and that forms the outermost layer of the testis. So you can appreciate the tunica albuginea here. Here we can make out the serous sac, which is tunica vaginalis, or we can at least appreciate as the external part, the outermost covering of the testis, as the tunica albuginea. Okay, this albuginea is basically what is a very dense fibrous substance. Okay, let's take a look at it more closely. It's very dense fibrous tissue. Is made up of collagen fibers and of course some components of elastic fibers. I'm very intentional in trying to use histology slides to show you, because I know several of you are going to be working on histology of the testis, and you are supposed to actually be able to appreciate your slides when you see them, be able to identify some of these components and know what exactly they are meant for. So you have presence of collagen fibers, you have elastic fibers, you have also the tunica albuginea. Like I said, is thick. It has collagen fibers, fibrous It's fibrous, of course, and then there is a bit elastic in nature. It convolutes together and then forms what we call the mediastinum testis. You can appreciate the tunica albuginea at this point and how they tend to send septa into the main tissue substance of in fact, the syncytium of the testis, and then begins to divide them into lobules. So when they send this septa within them, they divide the testis into lobules. I was about telling you the three different layers of the coverings of the testis. The external part, we talked about the tunica vaginalis, you have the tunica albuginea, and you have another one that is called the tunica serosa. Okay, I can't, it can't be made out here, but there is a third layer here as well. So, but the one that is related to what we are talking about is tunica albuginea, which forms the outermost covering of the testis. Okay, the mediastinum, as you've seen it, is a conglomeration of tunica albuginea, and then at some point where it forms straight tubules, they unite together to form what we call the erected testis. At this point, they unite together as straight tubules. They no longer form convolutions. They become straight, more like there is termination of it loses its convolutions at that point. Okay, it loses its convolution at that point. Remember that I said that the essence of the tunica albuginea is that because they are dense fibrous substance, they move into the syncytium of the testis and divide them into lobules, and then forms what we call different convoluted seminiferous tubules. So each of the lobules contain, because of that division, each of the lobules is contains one or more, usually between 1 to 3 tubules. Okay, and that is why we say that each testis has about 200 lobules and about 400 to 600 tubules. Let me say that again. Each testis has about 200 lobules and about 400 to 600 tubules. 400 to 600 because you have about 2 to 3 convoluted seminiferous tubules within one lobule. Okay, so each tubule becomes continuous with others as a straight tubule, which we call the tubuli recti. Each tubule becomes continuous with others as a straight tubule, and then at some point they form together. There's some point where they form the rectus testis, where they form together as straight tubules and form what we call the rectus testis. Okay, so you have also other things present within the space of each lobule. You have the loose connective tissue. You can see the connective tissue. You also have blood vessels, presence of blood vessels, which we can't make out here. And you also, of course, have lymphatics. And usually lymphatics flows alongside the same line of flow of the blood vessels as well. So we take a look at the seminiferous tubules. Now within each lobule, we want to focus on the seminiferous tubules. So now you can see, like I said, you're going to have 2 to 3 of them, within a particular lobule. Okay, so in each of the tubule, you are going to see that the rounded shaped thing forms a particular tubule, and within that particular tubule, a lot of things go on within it. It contains layers of cell. In fact, remember that I started by saying that the testis is where the sperm is being formed. Okay, after the germ cells, during the development, begins to move, and within the 4th to 5th week, and the germ cell has taken its position, okay, taking its position in the definitive organ that is supposed to form the reproductive system. at this point, remember that for male, the maturation starts happening, for female, everything, grows at the embryonic stage. And then during puberty, other maturation, second meiotic division gets to complete, and then other maturation complete. But for males, you discover that it comes at a resting position until puberty, when everything starts to, you know, uh, begin again. So seminiferous tubules, looking at it here, you're going to appreciate the fact that all the cells, all the sperm cells, or what I call the spermatogonium, the initial germ cell that is responsible for producing every damn sperm that is used in the body. So they are the spermatogonium, they are the stem cell, they are the germ cells, the ones that are definitive to form sperm. Okay, you see them arranged in a very orderly manner. They are arranged in layers, so when you come to the seminiferous tubules, what you're going to notice is that you're going to have layers of cells that are arranged in order of the growth of the cell, from spermatogonium to first spermatocyte to second spermatocyte, and then to spermatid, and then to full grown spermatozoa. It also contains smooth muscle-like cells, or what we call the myoid cells. You can appreciate the myoid cells, which forms usually lies at the basal lamina. Now appreciate this boundary. This circular boundary I'm showing you right now is actually where the boundary of this particular tubule is, and besides it, you're going to have another tubule, trying to form just at this point. So at that initial boundary layer of the tubule, you have the myoid cells. Okay, the myoid cell lies just at the basal lamina of the seminiferous tubules, and then just immediately after that, you're going to appreciate other levels of other the different germ cells of the sperm at the different growth stages. You can see the spermatocytes, you can see the full grown spermatozoa. I think this is actually not spermatozoa, this is the spermatid, even though the magnification is low, but I can make out that it is actually a spermatid rather than a spermatozoa. Within the lumen, that is where you likely will find the full-blown sperm cells. Okay, the layers of cell include the germ cells. I've told you the germ cells are the spermatogonium at different stages, and also primary spermatocyte, secondary spermatocyte. Those are the germ cells. And it also contains what we call the Sertoli cells or substanticular. So we're going to see it later. These sustentacular cells or Sertoli cells are actually, they play a supportive role, give a supportive role. They form a kind of tie junction system, that gives a supportive role to the germ cells, to enable them, to nurture them, to also enable them. They also produce what looks like an androgen substance that supports the growth and development of the germ cells. Okay. And then you also notice that there is lack of lumen or cell boundaries within the seminiferous tubules. Okay, there is not really a, each the cells the cells are all clustered together. You can see all the spermatogonium cells all being mashed together, and when you slice them, let's say you are taking a slice shot at different layers, you're going to find different cells all mashed together, usually without boundaries. And then when you also look at the lumen, within the lumen of this seminiferous tubules, you also find out that there is no boundary. You can't really say that this is the boundary, because a lot of things can be found there that there seems not to be a boundary, but of course you'll be able to make out the presence of lumen. So the myoid cells are responsible, for contractile movement of these germ cells within these different layers. Normally, the people have described different scientists have described about 6 layers you can find, and those 6 layers correspond to about 3 layers that contains the spermatogonium, and about one layer for the first spermatocyte, another layer for the second spermatocyte, and then another layer for the spermatid. So that corresponds to 6 layers. So the early, the first 3 layers are the early stage of the germ cells, where you have the spermatogonium. We're going to see the different types of spermatogonium, the 3 different types, and then those 6 layers are what you will likely find in any slice of a seminiferous tubule. Okay, let's now look at the spermatogonia or spermatogonium. Okay, so let's look at it. Of course, you already know the developmental stages, where you move from spermatogonium, to different types of spermatogonium, or the germ cells. We have what we call the type A, the type AD, or the type A dark spermatogonium. Okay, this is more like the stem cell. The stem cell backup. They form the stem cell, the real base, for renewal, to ensure that the spermatogonium doesn't get to finish. So they form a baseline. While the division is also happening, they also form a baseline to replace themselves. So that is why we say they are the stem cell. The germ cell responsible for making sure that the germ cell is being replenished. Okay, so we have the type A of it, and that type A have 2 types. We have the dark one and we have the pale one. So they sometimes see it called type AD or type A light. AD means dark, and then AL means A light. So we have about 2 types of them, type AD and type AL. And when they divide by mitotic division, they also form more of the type A dark cells, and then very few of the type the light. So they stain dark actually. Those that are called type A dark stain dark, and then those that are called type A light, they stain light. We also have the type B, the type B spermatogonium. The type B spermatogonium, you can appreciate that at this stage. The type B spermatogonium are the ones that stain light as well, and they produce more of itself and produce more of light than they produce dark. So they are basically stain light, and then they produce itself by mitotic division. At this level, we are still having the mitotic division of the spermatogenic process. Remember, you have mitotic divisions, and then you have first meiotic division, you have the second meiotic division before the production. Remember that the essence of the spermatogenesis is to establish the haploid number. Remember, because you need to have a haploid system. It's coming from the human body. The cells are coming from the human body, so they are diploid in nature. The essence of meiotic division is to make sure that they reduce the number of chromosomes from 46 to 23, such that it will become a germ cell. It will become a sperm, which will have 23N, which means the haploid number, such that when it meets the egg also having 23N, be able to produce a zygote, which will now reestablish the human diploid chromosome number. Okay, so the spermatogonium forms the spermatozoa stem cells, and one of the other things you're going to use to differentiate between the type A and the type B cells of this is that the type A will have an oval nuclei. So the nuclei is usually oval in shape, while the nuclei of the type B is spherical in shape. This is a character you may not be able to make out this in a typical class slide, okay, but under electron microscopy, you should be able to appreciate this and then see the variations within the nuclei. And then you also discover that, remember I said that the growth stage from the beginning lies at the basal lamina. So the spermatogonium naturally, type A, type B, all the 3 stages are going to lie at the basal lamina, and they give rise to primary spermatocytes by mitotic division. So you can appreciate them here. This is the H&E stain of testis, and you're going to, within this seminiferous tubule, you're going to appreciate the spermatogonium Look at this one. Okay, and within each of them, you're going to also appreciate the fact that how the Sertoli cells are being sandwiched within them. Okay, and then just after the tunica, you can see the red thing, the very thick red thing is the tunica albuginea, okay, which is the outer covering. And then after that, you're supposed to immediately see the spermatogonial cells, spermatogonia. You can see make them out, and then within each spermatogonia, you're going to be finding Sertoli cells being sandwiched within them. Also on the right side, the B photomicrograph, you're going to also appreciate the same thing. So from the tonical albuginea going down, inside into the lumen, you're going to find the stages in a progressive manner. You're going to find the layers and the stages of the growth stages of the sperm in a progressive manner. Now we move to the next phase, which is being formed by the spermatogonia, the primary spermatocyte. The primary spermatocyte is formed by a mitotic division of the spermatogonia and is actually the largest cell. Remember, the spermatogonium is larger and larger than primary spermatocyte. In fact, as it grows, and as the sperm grows, it reduces in terms of size. Okay, it reduces in terms of size. Even the nucleus also reduces as well. So there is the spermatogonium is large, and as it goes down, as it grows to primary spermatocyte, it becomes less, large, or it becomes smaller. So it means that primary spermatocyte is smaller in terms of size compared to spermatogonium. Note of that. I don't know, but more likely, I'm going to be asking us about. I'm going to be having some form of MCQ, for this course. I don't know, but it's what I'm thinking. Maybe before your exam proper. So take note of these tiny details I'm making mention of. So primary spermatocyte basically is formed by mitotic division of spermatogonia, and they have large spherical nuclei, but of course not larger than spermatogonium, and they give rise by they give this the time at which the first meiotic division gets to start. You can see the process that I illustrated here, the primary spermatocyte, and then to divide itself by meiotic division to form the secondary spermatocyte. Okay, so they reduce the number of chromosomes to half, because at that point, remember, it's still mitotic division. From spermatogonium A to spermatogonium B is a mitotic division, and from spermatogonium B to primary spermatocyte is still meiotic, so it retains its fixed number of chromosomes until meiosis comes in. This reduction is still there. Will still not happen. Okay, but at this point, primary spermatocyte begins to divide to form secondary spermatocyte, and then at that point, you would have established an N, forming two daughter cells, and then each of them being a haploid cell. So now you can also make it out. First of all, you have the tunica albuginea, and then you have the primary spermatocyte, and then you have the secondary spermatocyte. Now we are closing in towards the lumen, and then you are beginning to appreciate the different stages of that growth. Now the secondary spermatocytes also gets to undergo meiosis too to form the spermatid. Each of them forms two spermatids, and then of course remains the haploid number remains established. Okay. At this stage, nothing happens to the haploid number, during this meiosis. Okay, nothing happens. Nothing is lost at this stage. So what happens is that you have the DNA number also being refined. Okay, the haploid number has already been restored, and then we are having formation of spermatid. At this stage, we are having, sorry, I was saying the secondary spermatocyte gets to form two spermatid after it has undergone the second meiotic division. And then by then, I'm going to show you later how the what the spermatid looks like. I'm appreciating here, but it's not quite clear. I'm going to show you a clear picture of what it looks like. But then you can find the secondary spermatocyte at this point, and then you can also find the tiny spermatids also at this point, just very close to the lumen. Okay. Now the spermatid, what are they? The spermatid are more rounded. Remember, I said that the size begins to decrease, and then the cells begin to become rounder as the growth progresses. So they are rounded cells. They are smaller than spermatocytes, and of course, what basically happens is a change in shape and a change in organelle, the organelle orientation. So you're going to see a rearrangement where you have the nucleus coming to the apical part of the spermatid, and then forming what looks like an acrosomal cap, and of course you are going to have the nuclear condensation also happening, and then you're having the mitochondria arranging itself towards the microtubules. It will arrange itself towards the microtubules and then form what you call the middle piece. And then the joint between the place where the nuclear condensation happens to the middle piece becomes the neck, you know, the neck eventually, which becomes eventually the neck of the sperm. So these arrangements in shape, and of course there are many things that inspire this, that stimulate this at this stage, which is not part of this course. The basic thing is to show you the microscopic features and what you get to appreciate at the level of testis and spermatogenesis. Okay, eventually the spermatid gets to form the spermatozoa through the spermiogenesis process. This spermiogenesis is when spermatid is now being refined to become a full-blown and full mature spermatozoa. Okay, so I've talked about the change in shape, the organelle orientation, and then you're going to have the presence of the Golgi apparatus, usually sometimes being lost, and their mitochondrial arrangement in following the tubular, the what do you call it, following the microtubular structure in a more like a parallel line. Okay, this is what I'm talking about. You can see the mitochondrial arrangement at this point. You can see the nuclear condensation, and then you can see the acrosome coming to lie at the tip, to where eventually will become the head of the sperm. Now let's look at the specific microscopic events of spermatogenesis that converts the spermatid to a full-blown spermatozoa. I've talked about nucleus condensation, and then the Golgi complex, which transforms into the acrosomal cap. You have some of them being lost, and also some of them being utilized to form the acrosomal cap, and then lying just at the tip after the condensation of the nucleus at the tip to form the acrosomal cap. Of course, this is very important because that acrosomal cap is responsible for the future capacitation and acrosomal reaction, that will ensure that fertilization, takes place. Okay, there is also division and migration of the centriole and formation of the axial filament. That's what we call the microtubules, the centriole, and the gets to form the axial filament, which is the microtubules. So being formed at the middle part, and eventually having the mitochondria arrange itself, in a very in a parallel line along that tubular structure, forming the neck, separating the neck from the head, and also forming the middle piece. You can appreciate that a little bit more here at the G, A, F, D, E, F, G, at the G point. So you can see the central, okay, and then you also appreciate the mitochondrial sheet having arranged itself and formed the neck. Now you can see a separate neck. You can see the middle piece, what you call the middle piece. Now you can make out the head with the acrosomal cap. So while the Golgi complex forms the acrosomal cap, the central gets to align to be able to form the middle piece. Okay, and then we also have so all this part is actually the microtubular structure, which is formed by the central or the axial filament. Okay, and then you also have cytoplasm being shed to reduce the size at this point. Remember, it's the spermatid is round, is rounded in shape, is rounded, and then at this point, the presence of excess cytoplasm will be shed off. As it's being shed, is part of the shaping. Remember, I talked about nucleus condensation and changes in shape. So changes in orientation and organelle organization. So all this, as the cytoplasm is being shed, the shape is being refined to be able to have that pin looking shape of a spermatozoa. Okay, but of course, nothing happens to the cell membrane. The cell membrane persists and still forms the outer covering of the spermatozoa. Now let's look at the one of the last things, the cells that needs to be appreciated in this process, which is the sustentacular cells. The sustentacular cells uh, are cells that you find within or what you call the Sertoli cells. Actually, I've talked about in the beginning. We saw that as Sertoli in the beginning, and I normally like to call it sustentacular cells because they sustain. That way it helps me to relate with the function. So you can see, you can appreciate the way the cells are sandwiched, like I used before, just between the growing spermatogonium cells. So they tend to support more like it's more like a physical kind of support, you know, not necessarily functional or physical, but of course there are evidence to show that there's some liquid is being, some substances rather are being secreted by this particular cell, which are useful for the growth of spermatogonium cell, at least that they be not lost. Okay, so you see them, you can see the shape, they are irregular in shape, they are pyramidal, they look like what you see also in the feminine when you do a vaginal smear. Okay, you can see that irregular shape, pyramidal in shape. They're also tall, being sandwiched, more like offering some level of protection to the Sertoli cells. Okay, the nucleus lies at the base. You can appreciate the nucleus. By the way, this is not a histology slide. This is not a micrograph. This is just a made-up diagram to show you, to depict the histological positing of some of the cells. It has the prominent nucleus which lies at the base, and you have it being interposed. You have an interposition of the spermatogonium around them. Okay, so it supports developing germ cell and supply its nutrients, and usually acts as macrophages to give some level of protection. You know what macrophages does? You know, they remove infections, they fight, you know, foreign bodies as well. They try to, you know, eat them up and then digest them within. Now, they are less prominent, you know, than germ cells in adults. So in adults, they become less prominent. Okay, so the germ cells become more prominent in adults. Of course, you know the reason, because at puberty, once you have reached puberty, of course, the activities increase, because at that time the germ cells, you know, begin to proliferate even more. So you find out that the sustentacular cells becomes less prominent in adults, but become more prominent in younger individuals. So we are going to see that they connect to each other by adjoining, you know, tight junctions. You can see the relationship it makes with itself and with the cells surrounding it by tight junctions. And then it has a presence of cytoskeleton within it that are very important for making that cohesive function. These cytoskeleton that are found within the spermatogonia cells are probably what, you know, enables it to, you know, give the level of support it gives for the spermatogonium cell, the germ cells, and they produce a substance called the Mullerian inhibitory substance. Okay, and they also produce androgen binding protein, and also they produce inhibin. But of course, it's not exclusive to them, but it's part of the things that they produce. Remember that Mullerian inhibitory substance, if you remember, is a hormone that is responsible for making sure that the Mullerian body, does not, doing. Remember, is the Mullerian body is responsible for determining who is male or female during the growth stage development of the embryo. So once you see that this is produced at for a particular embryo, once Mullerian inhibitory substance is there, it inhibits the Mullerian body from being from developing. So it means that that particular embryo eventually becomes a male. Okay, but when this MIS is not present in a particular embryo growing fetus, what it means is that Mullerian body is definitely going to be produced, and that embryo will turn to be a female eventually. Okay, so that's the role of testicular cells. And it also produces androgen binding protein, of course, to be able to help even for the production of testosterone. That's one of the roles of androgen binding proteins, and facilitate the production of testosterone, and of course inhibin as well for feedback mechanism of reproductive hormones as well. Now let's look at the maturation proper. Maturation of spermatozoa. What happens is that when the spermatozoa we've talked about how it comes up to its latest stage from spermatid, undergoing some spermiogenesis, cutting off some level of cytoplasm, rearrangement of the organelles, and then the central forming the middle piece, and then you are having arrangement of the neck, and then you are having the acrosome, the Golgi apparatus forming the acrosomal cap condition of the nucleus, and so on. Eventually you form a spermatozoa. What happens? And then they begin to move. They move to epididymis, where they get stored, right? And then at that level of epididymis, they get some constituents that also try to make them up, that make them what they are, the real spermatozoa that should be active. But then they are not yet active at that point, until they are ejaculated into the female tract. Okay, at the level of epididymis, we see that is what we have some surface molecules that get attached, that are actually attached at the epidermis, are more like they mask these sperm cells. We call them, they are proteins, a mixture of proteins and carbohydrates. This is what is illustrated by the yellow thing you are seeing right here at this epidermal level. So it forms a kind of complement of surface molecules, a mixture of proteins and carbohydrates, and mask these sperm cells. And um, of course, at this point, you would have had a mixture of within the semen now, not just the sperm. Remember, sperm is just the 5% of the makeup of any ejaculate. You're going to have the mixture from the seminal vesicle. You're going to have from bulbar urethral gland, maybe about 5%, and going to have which other one? Remaining one contribution again that gives you another fluid content into the makeup of the entire semen. So, but this sperm does not get capacitated, does not get matured fully until it's ejaculated. It's until ejaculation that you begin to have further molecules again, by activation of other components within the female tract system. Will, now you can see a little bit these molecules that cover the acrosomal cap, get other molecules, masking them again as a result of being released into the female tract. And that release into the female tract, of course, is, elicits some level of responses that do what we call capacitation, that respond to what we call capacitation. So that capacitation is the first ability that the sperm possesses, okay, that makes it ability, that gives its ability to fertilize an egg. Okay, so those substances that mask the acrosomal cap, will be broken down, and they are going to be exposed. Once the acrosomal cap begins to be exposed at that point, so that it can now have freedom to interact with the egg, capacitation is now happening. Okay, capacitation is now happening. Of course, there are chemoattractants that elicit these responses, and of course, there are also some. Remember that zona pellucida that covers the egg also is a glycoprotein and also releases some enzymes as well. And then there are also enzymes that are also being released by the acrosomal cap itself, and these enzymes are lysosomal in nature. Okay, and one of them is hyaluronidase. This hyaluronidase is basically to be able to clear out all the corona radiata, to digest them and to make way for the entrance. If you appreciate the second picture here, you can see at this stage the zonal reaction is happening, where you have the zonal corona radiata being destroyed by hyaluronidase, and then further release of trypsin-like substances, and of course the acrosin, which enables the burrowing of the egg by the spermatozoa. Remember that the egg is a glycoprotein coat, and we also need some level of reaction to be able to break through the bilayer and then to move within the perivitallin space of the egg. And once that happens, and the first sperm is able to do that, it by that zonal reaction, it locks up the entire membrane of the egg, so that another sperm will not be able to enter as well. Okay, so all of these processes leads to what we call the maturation of the spermatozoa. So if a spermatozoa does not, remember the motility of the spermatozoa, remember we talked about the presence of myosin-like substances, smooth muscle myosin-like substances that are found within the seminiferous tubules that help to move them as they grow. So the contractile activities of these muscles, they move these sperm cells, at the level of spermatogonium, at the level of primary spermatocyte, at the level of secondary spermatocyte. They propel them, and they move them towards the lumen, such that when they have become full-fledged spermatozoa, they can move to epididymis for storage, right? At epididymis, they also gain some molecules, and they become a little bit, have some level of mobility, but they don't become motile until they are being released into the female trial. Once they do that, you discover that by also chemoattractants and some molecules that are also within the feminine, they even move beyond the motility they have obtained at the epididymis. They move beyond that because there are also even microvilli that is present in the pathway, the uterine pathway, also enable them to move faster. They actually move faster than their normal motility to reach the egg as well. So all of these make up the motility and cause capacitation and zonal reaction that is seen in the growth of spermatozoa. Now we look at the last thing, the interstitial cells. The interstitial cells, from the name, is interstitial. It lies between, it's kind of a switch. It finds itself, between two things. It lies between, two lobules, or two tubules. You find it more like standing at the middle point, communicating between 2 to 3 seminiferous tubule. So you can see this tubule right here, you can see this one right here, you can see this one right here, and then you can see within that communication, you are going to see the interstitial cells, or what we call the interstitial cells of Leydig. Some people say Leydig cell, but the real name is, the English name is interstitial cells. Leydig is just the name of the person that discovered it. So we normally say interstitial cells of Leydig or interstitial cells. Okay, so you can see the cells themselves. They are also round, large, and polyhedral in shape. Their nucleus are eccentric. You can see eccentric means that they are towards the equator. Now you can see the arrangement. Let me just appreciate this one, this particular one I'm pointing at. You can see the nucleus, that very dot, mauve color thing is a nucleus. You can see how eccentric it is, almost towards the end point of that line. Okay, sometimes cytoplasm stains light with foamy appearance. Foamy appearance because of the presence of lipid. So it has some level of lipid present in it, and then you begin to see some level of foamy appearance, when it is being stained. And you also have some presence of yellow granules. This you see, and lipofuscin, of course, as a result of lipid as well. This you see when you look at it, under the microscope, with the EM, electron microscope, not just the light microscopy. And then you have endoplasmic reticulum that does not have granules. Okay, so it's clear. And we have some rod-shaped crystalloids that are present within the cytoplasm. All these things are things, but these are microscopic features that you need to appreciate that makes it perform its function. Remember that these cells are critical because they are responsible for uh, producing the testosterone, right? They secrete the testicular androgens, whether it is dihydrotestosterone, the one that is not yet inactive, then that will be activated to the testosterone. So they are the real deal. At that junction where they situate themselves between the seminiferous tubules, they are responsible for producing testosterone, and of course by the stimulation from the luteinizing hormone of the pituitary gland. Remember, luteinizing hormone is also otherwise called the interstitial cell stimulating hormone, right? So that hormone, from the hypophysis cerebri, coordinates, or rather regulates the function of the interstitial cells that makes it to produce the testicular androgens. Within these cells as well, you also find presence of collagen fibers. You also find presence of fibroblasts, macrophages, mast cell, blood vessels, and of course, lymphatics. Okay, we're going to stop here, and we look forward to discussing that of female. We're going to look at hopefully uterus and ovary, and also try to look at some of the reproductive cycles that is characteristic of the females, and look at the microscopic features that are relevant that we should focus our attentions to. Okay, thank you very much, and you can use my emails to send me any feedback, any questions, or hopefully if I have a next class physical, be able to have interactions on this, and I look forward to a quiz exam regarding this particular topic. Thank you.